Good afternoon, Pratham. Okay. So I'm Rahul Saha and we'll be talking about the last few questions which were left, okay, but a lot of concepts were remaining in that chapter. Okay, so we'll be discussing regarding about that. Yeah, good afternoon. I'll be discussing that concept and explaining to you how to solve the questions regarding the excretory tract or the urinary system. So in this type, in this one, subjective test also I have put the excretory system because it has been taught. Okay, so in that worksheet, let's come to the question number. You can say 26. Till 25 we had done. So come to the question number 26. If Henry's loop were absent from the mammalian nephron, which of the following is to be expected? There will be no urine formation. There will be hardly any change in the quality and quantity of urine. The urine will be more concentrated. The urine will be more dilute. Okay. So the answer looks quite easy. The urine will be more dilute, right? But how it will be more dilute, the answer is in the counter current mechanism. Okay, the counter current mechanism which occurs as per, good afternoon Arya, which occurs as per the counter current mechanism, and counter current mechanism has two states known as counter current multiplication and counter current exchange. Arya, just look at question number 26. That application of that question is counter current mechanism, that how urine will be more dilute. The answer is urine will be more dilute, but you should know the explanation part, right? So the thing is, how that is done, I'll explain it to you now, because each question's explanation you should be knowing, okay, and it might not have been taught to you also. So counter current mechanism, when you talk about, okay, so how does it happen? So the thing is, first of all, you should know something, that this specific countercurrent mechanism, the nephron loop and countercurrent multiplication, okay, is established in the loop of Henle. So the kidneys couple two countercurrent mechanisms to establish the condition necessary to regulate the volume and concentration of urine. Okay, regulate the volume and concentration of urine. Listen to this extremely carefully, as this concept is very very important, and directly this concept is applied in that question. Okay, then after we have learnt about this specific stuff, then what we can learn more is this counter current mechanism is basically made up of counter current, you can say, multiplication and counter current exchange. Okay, counter current multiplication and counter current exchange. But what does counter current means or mean? Counter current refers to the fact that the exchange takes place. Counter current, what does it mean, counter current? Okay, so be very very basic about it. It means that the exchange does take place, okay, between fluids moving in opposite direction. Exchange means exchange of solids and water does take place between the, you can say, fluids moving in opposite directions. Opposite directions, you can say, in two adjacent segments of the same tube. Okay, two adjacent segments of the same tube. I'll make it clear to you very shortly. Just of the same tube. I'll draw the loop of Henle here. Okay. Now, these two mechanisms are known as countercurrent multiplier. Okay, because the filtrate flow in the nephron loop will multiply the concentration and countercurrent exchange, that blood flow in the vasa recta, that I'll explain to you. Okay, so what happens is, now I'm drawing the loop of Henle for you. Okay, or I'll rather do this one. I'll rather share a screen, that will be much better. Okay. Okay, I'll just share this screen. Let me just tell me whether you are able to see it or not. Okay, now tell me, can you see the screen? Can you see the screen? Now you'll be able to see this screen, and now look here. Okay, I'll explain from here only. So the nephron loop and countercurrent multiplication. So the descending thick limb and the, you can say, thick ascending limb of the nephron, they lie very close together. Okay, they're separated only by the peritubular fluid. That's all, which surrounds all the nephrons. Okay, which surrounds all the nephrons. So they lie very, very close to each other. Now the exchange of substances, you can look here. Okay, peritubular fluid is this one. Okay, this one is the peritubular fluid, which is separating the ascending limb, thick ascending, and thin descending or descending limb of loop of Henle. So only this peritubular fluid is segregating the two parts of the nephron, which is present in, it surrounds all the nephrons. Okay, now also I told you that the thick ascending limb doesn't mean it is very, very thick. It is not stratified. It is just cuboidal epithelial cells. Here this is simple squamous epithelial cell in the thin limb or the thin ascending limb. Okay, this is a thick ascending limb present in the longer loop or the medullary nephrons. Okay. Now coming to further explanation, what happens is, the exchange of substances, okay, between these segments of a nephron is known as countercurrent multiplication. Means the tubular fluid in the descending limb, it will flow towards where? It will flow towards the renal pelvis, while tubular fluid in the ascending limb, it flows towards the cortex. Now multiplication refers to the fact that the effect of the exchange increases as movement of the fluid continues. That is the meaning of countercurrent multiplication in countercurrent mechanism. Now what is the function of countercurrent mechanism here? The countercurrent multiplication performs two functions, or it will perform two duties, you can say, or functions. Number one is, it efficiently reabsorbs the solutes and water. It efficiently reabsorbs the solutes and water. Before the tubular fluid reaches the DCT and the collecting system. Okay. Before the tubular fluid reaches the DCT and collecting system, or collecting Now, it will establish a concentration gradient in the peritubular fluid also. Establishes a concentration gradient in the peritubular fluid. Okay. So when it does establish a concentration gradient in the peritubular fluid, it is known as the medullary osmotic gradient that permits the passive reabsorption of water from the tubular fluid in the collecting system. Now this will allow the production of concentrated urine. Okay, means the urine will become more concentrated and more if ADH is again, you can say, secreted and causes more water reabsorption. Now there are two parallel limbs I told you of the loop of Henle or nephron loop, which have very different permeability characteristics. Okay, so the descending limb is permeable to water. Know it very clearly, like carefully, and it is impermeable to solutes. Very carefully note this fact. Okay, descending limb of the loop of Henle, the descending limb of loop of Henle here it is impermeable to solutes but permeable to water. Permeable to water. Very carefully. Okay. But the thing is, it can contain active transport channels which will pump sodium and chloride ions from the tubular fluid into the peritubular fluid of the medulla. That it can do. Okay. Now, look at this figure. What happens in this figure is the sodium ions and chloride ions are pumped out of the thick ascending limb. Okay. And before that, I have just forgot to tell you about the thick ascending limb. The thick ascending limb is relatively impermeable to both water and solutes. Okay, the thick ascending limb is impermeable to water completely and somewhat to the solutes, but it does contain some active transport system for solute taken, like the Na plus K plus 2Cl minus important that it contains, but it cannot let water pass through, right? It cannot let water pass through. So the descending limb is permeable to water, but relatively impermeable to solutes. Okay, and so you can write here relatively. Whereas the thick ascending limb is relatively impermeable to both water and solute, but can contain active transport mechanism that pumps sodium and chloride ions from the tubular fluid into the peritubular fluid of the medulla. Okay. Now the sodium ions Na plus and chlorine ions Cl minus are pumped out of the thick ascending limb. Okay, into the peritubular fluid. Okay. Now look at this diagram. When they are pumped out into the peritubular fluid, you can see the arrow. Okay, via the Cl minus leak channels, and you have Na plus NAK ATPase. So they are pumped out. Okay, when they are pumped out, what you are seeing is, you can see 1200, 900. So this is what? This is the osmolarity of the urine, the filtrate. Now what is happening is the water concentration is increasing when you are pumping it out. So the urine is becoming more dilute and osmolarity is decreasing. Here water concentration or solvent concentration is increasing and osmolarity is decreasing. Urine is becoming more and more dilute here, right? So once the urine has become more and more dilute, point number two will be what? Point number two will be this pumping action increases the osmotic concentration in the peritubular fluid now, right? The peritubular fluid will have increased osmotic concentration or not, right? This will increase the osmotic concentration of the peritubular fluid, right? Because you are throwing or pumping out ions, or you can say solute, into the peritubular fluid from the tubular fluid. So in return, what will happen is this will create a small concentration difference between the tubular fluid and the peritubular fluid in the renal medulla. Okay. Now the concentration difference results in an osmotic flow of water out of the descending thin limb into the peritubular fluid. Now see, the descending thin limb will throw out water in order to compensate this increase in the osmotic concentration in the peritubular fluid, which happened because of the ions being pumped out from the thick ascending limb of the loop of Henle. Now look at this as a perspective. Okay. Now when you have looked till now, then what will happen now is, as a result, the solute concentration of the tubular fluid, okay, in the descending limb will increase or decrease. It will increase because water is coming out. Now the arrival of the highly concentrated tubular fluid in the thick ascending limb, okay, will speed up again the transport of sodium and chloride ions into the peritubular fluid, because the water would again be too much highly osmotic and it will throw out because its concentration will be, you can say, more than the peritubular fluid, this osmolality, and it will again throw out, and this cycle will keep on continuing, keep on continuing. So it's a kind of multiplication. So if this does not happen, what is happening? The water is not getting reabsorbed. Okay, from here the water is not being thrown out, and then the urine will be very, very dilute later on. Okay, so this is half of the countercurrent mechanism. As per that, that question was, you can say, put on. Then the next question, question number 27, come to that. Conversion of ammonia to urea is done by the urea cycle, also known as the ornithine cycle. Okay, it is also known as the ornithine cycle. Question number 20, I think 7. Okay, so I had shown you the ornithine cycle once. Okay, I will show it to you again. Let me. So this ornithine cycle, it takes place, urea cycle takes place in the liver and converts the ammonia present in the liver, okay, or the ammonia achieved, or it has been received from the, or after the deamination process that happened in the, or that occurred in the amino acids. Okay, so deamination of amino acids which occurs in the liver by transaminase molecules, okay, or transaminase enzymes, that transamination or oxidative deamination, okay, that causes ammonia to come out, and that ammonia is converted to urea, okay, via the urea cycle. Okay, so it is a long cycle, okay, present in the mitochondria and your, you can say, endoplasmic reticulum, sorry, mitochondria and cytosol both. Okay, so it is a shuttling reaction, you can say, the urea cycle. So that is also known as the ornithine cycle. Okay, the urea cycle. So you have carboyl phosphate that becomes your, in the end, you can say, citrulline becomes arginosuccinate. Okay, I'll just show the urea cycle to you, and then ornithine comes back. Okay, first it will react with ornithine, the urea cycle. Look here, you have carboyl phosphate synthetase, okay, type 1, okay, CO2 plus NH3 plus ornithine, transcarbamylase ornithine will come in. Okay, this requires two phosphonitrile bond dissociation, ATP becomes AMP plus PPI. Then you can say this synthetase process I was saying about. I was talking about the first process. Then you have transcarbamylation. This ornithine comes into the mitochondria. This is the mitochondria. Citrulline, aspartate comes in. You have again a synthetase requiring ATP. Then you have argininosuccinate. Then you have the succinase. It will break it down. Fumarate will come out, which is a Krebs cycle intermediate. Okay. Then arginine, and now again arginine will be broken down. Okay. Urea will come out. Again ornithine, and the cycle will keep on going. So this is how you release urea. You convert NH3 from here to urea here, and you excrete it via the DCT. Okay. So next question. Concentration of urine depends upon which organ? Okay. So that is again the concentration of urine. Question number 28. So it depends upon, again I told you, again counter mechanism, the longer it is, the more concentrated your urine will be. That is the length of the Henry's loop or loop of Henle. So if you have longer loop of Henle, you will urinate lesser and lesser, just like in camels. Okay, evolutionary perspective, because they cannot afford to lose a lot of water because they have a huge loop of Henle. If we had that much long loop of Henle, medullary loop means a longer loop of nephrons which have more thick ascending limb, so we would be setting up independent regulation like we do still, because thick ascending limb is impermeable to water and solute, and mostly little bit permeable to solute, but descending limb is impermeable to solute, but you can say permeable to water. So we are setting up independent regulation of water and you can say solute, so that the concentration can be enhanced of the urine, and water can be reabsorbed more and more. So if you want to reabsorb more and more and more water, like in case of desert animals, you can increase the length of the nephron loop, also known as the loop of Henle or Henle's loop. Okay. Now question number 29. A condition of failure of kidney to form urine is known as, question number 29, that is known as anuria, no formation of urine. Okay, anuria, no formation of urine. Deamination is, I told you what is deamination. Okay. Then uremia is, you can say, a condition which involves high level of, you can say, waste products, okay, in the urine. Okay, but like you have blood urea, nitrogen, creatinine, but uremia will lead to anuria or will lead to kidney damage, but here it is saying no urine formation, that is anuria in the end. Okay. Now the basic functional unit of human kidney is the nephron. Okay, we have talked about the nephrons. Okay, so the nephrons are 1.25 million in each kidney. Each kidney has 1.25 million nephrons, and each nephron is made up of renal corpuscle and renal tubule both. Okay, and you have something known as, there are two types of nephrons also. Okay, if you are not aware of it, there are two types of nephrons. One is known as the cortical nephron, one is known as the juxtamedular or medullary nephrons. Okay, so 85% of the nephrons, 80 to 85, okay, are cortical nephrons. This question also comes in NEET examination, are cortical nephrons. Now when 80 to 85% are cortical nephrons, they are almost located entirely within the superficial cortex of the kidney. Okay. Cortex of the kidney. Next is, in a cortical nephron, the nephron loop is also relatively short. Short means it may not have the, or it does not have the thick ascending limb. Okay, and the efferent arteriole delivers blood to a network of peritubular capillaries which surround the entire renal tubule. Okay. Now when we come to the remaining 15% of the nephrons, they are known as juxtamedullary nephrons. Okay, they have long nephron loop rest 15% to 20%. Okay. Juxtamedullary nephrons. So they are very essential juxtamedullary nephrons in the sense that they have long nephron loops. Long means they have an additional thick ascending limb, okay, and they extend into the deep into the renal pyramids of the medulla. They extend deep into the renal pyramids of medulla. Okay, so very important. That is the nephrons. And the efferent arterioles of juxtamedullary nephrons they directly connect to the vasa recta, okay, the blood vessel, which is the long straight capillaries that are parallel to the, you can say, loop of Henle. Okay. Now the next question. In ureotelic animals, okay, urea is formed by, okay, so in ureotelic animals, urea is formed by what? Urea is formed by again the urea cycle known as the ornithine cycle. That is the same answer, the ornithine cycle. Now again coming back to the ornithine cycle, it removes two waste products from the blood in liver. These two are what? They are CO2 and ammonia. If you see this ornithine cycle again, okay, just look at this ornithine cycle again. What is this removing? CO2 plus NH3 plus you have one ATP, but two phosphoanhydride bonds are broken, so it is counted as 2 ATP energy. So it is broken down into AMP plus pyrophosphate PPI, and then this carbamoyl phosphate synthetase 1 produces the carbamoyl phosphate. And here what is being condensed? Two are added, like CO2 and NH3 plus are both added, or NH4 plus I should be saying. They are both added, ammonium ion and CO2. And when they are added means both are removed. Both are added and carbamoyl phosphate is formed, and carbamoyl phosphate is reacting with ornithine in the mitochondria. Why ornithine transcarbamylase? It is forming the citrulline compound which exits the mitochondria, comes into the cytoplasm or cytosol. This entire cycle goes underway, right? Arginosuccinate, then arginine, then again urea comes out. When arginine is broken down by the enzyme, arginase, ase is a suffix which means breakdown. Okay, it will break down the substrate. So substrate here is the arginine. Ornithine is being produced, urea comes out. Okay, so here it is removing both CO2 and NH4 plus means carbon dioxide and ammonia. Okay, so this is the correct answer. And this is a two-stage process again. No, half of the process or less than half of the process starts in the mitochondria, comes into cytosol, and then again this ornithine settles back into mitochondria, and this cycle keeps on running. Okay, this cycle keeps on going. It's a circular cycle, just like a Krebs cycle, keeps on running round and round. Okay, so that is the meaning of ornithine cycle, because ornithine is very important. It is also known as urea cycle because it throws out urea as a byproduct. Okay, so coming on to the next question. Which one of the four parts mentioned below does not constitute a part of a single uriniferous tubule? Okay, very good question. Okay, the question is, and the options are, which one of the four parts mentioned below does not constitute a part of a single uriniferous tubule? Okay, so options are distal convoluted tubule, collecting duct, Bowman's capsule, loop of Henle. Okay, so uriniferous tubule is what? They are nothing but nephron. Okay, so what is not present in a nephron? Just think. Just think what is not present in the nephron. So renal tubule will comprise of what? Proximal convoluted tubule, loop of Henle, and distal convoluted tubule. Renal corpuscle comprises of the glomerulus and that one, but renal collecting duct is present in the inner region of the kidney, which is the renal medulla. It carries the urine from the nephron to the or from the nephron in the cortex to the pelvis in the kidney. So it is not the part of one nephron, okay, but it is formed with the distal part of many nephrons, okay. Many nephrons they comprise together to form a collecting duct. So a nephrons, that is the collecting duct, will be the answer. So you can write the parts of a nephron here also. If you look at a nephron, it's made up of renal corpuscle, capillaries, and glomerular slash Bowman's capsule. Then renal tubule. This is proximal convoluted twisted tubule, then loop of Henle or nephron loop, also known as nephron loop. Then DCT, distal convoluted tubule. So this nephron is only the other name of nephron is nothing but uriniferous tubule. Okay. Uriniferous tubule. This is the correct answer. Okay, so collecting duct is not a part but is produced from or emanates by the, you can say, addition or the amalgamation of various DCTs of various nephrons, right? So coming to the next question, if excess water passes out from the tissue without being restored by the kidneys, if excess water passes out from the tissue without being restored by the kidneys, the cells will do what? Okay, so the what will happen is the loop of Henle, it creates a very high concentration gradient that I told you, okay, of salts in the tissue fluid in the medullary region or in the medulla of the kidney. So this causes a lot of water. The loop of Henle causes a lot of water to be reabsorbed, right? To be reabsorbed. Know it very, very carefully, to be reabsorbed from the fluid in the collecting duct as it flows through. Now producing very concentrated urine and preserving water for the body. So in the water, or you can say in the absence of this process, if the that question, the first 26th question, the first of the day that we discussed, if fluorophylline is taken out, what will happen? The water will not be reabsorbed, and then what will happen? The cells would directly shrink, and ultimately necrosis or cell death will occur. Okay, that will be the correct answer. So we'll so coming to the question number 34, this is okay. So it will shrink and die. Okay, so the glucose is taken back from the glomerular filtrate. Okay, again a good question. Glucose is taken back from the glomerular filtrate by active transport. Okay, mainly secondary active transport. Glucose is taken back from the glomerular filtrate. Especially in the PCT, what you have is 2Na1 glucose importer. So if this is one cell of PCT, these are the microvilli. Okay, this is the basolateral membrane. or basolateral side as we'll be saying. This is the apical side, apical membrane. Okay, this is the tubular fluid slash lumen from where the filtrate comes out. This is the inside of the cell. This is where the peritubular capillaries and peritubular fluid lies or lie. Peritubular fluid lies. Okay. Peritubular fluid. Now what we have is a symporter here is present secondary active transport which will take in 2 Na and 1 glucose. Okay, so Na comes down its concentration gradient, okay, glucose comes up its concentration gradient. So this is a secondary active transport. One, when it is coming down its concentration gradient, one of the molecule which is Na plus, 2Na plus, so it will bring glucose against its concentration gradient, okay, by harnessing the energy of its own potential energy, by harnessing its own potential energy which is coming down the concentration gradient, and then glucose exits via facilitated diffusion, and Na plus exits via, you can say, Na plus, K plus, ATPase present on the basolateral membrane. Okay, so this is how glucose is reabsorbed in the PCT only. This is also known as SGLT2 transporter. Okay, for glucose. Okay, very important. For again, there are various drugs in the market which inhibits this transporter, blocks this transporter, so that glucose is thrown out of the or through the urine, or glucose is thrown off the body through urine and reduces, and which will then reduce the blood glucose in the diabetic patients. It is also used in the treatment of heart failure. Okay, a person who are diabetic and suffering from heart failure, congestive heart failure. So next is question number 36. So nitrogenous waste products are eliminated mainly as, question number 36. So urea in the tadpole, ammonia in adult frog, ammonia in tadpole, urea in adult frog. Okay, urea in both tadpole and adult frog. Okay, urea in tadpole and uric acid in adult frog. So the answer is, you know that adult frog excretes urea, so it's a uricotelic creature, but a tadpole is an ammonotelic creature. So an answer is B. It is known as ammonia in tadpole and urea in adult frog. Okay. Under normal condition, 37, if you see, which one is completely reabsorbed by renal tubule? Uric acid, urea, salts, glucose. What it could be? The answer is glucose. Here how and why I'll explain it to you. We had just talked about the SGL2 transporter in the PCT. Capital side or membrane, you can write anything. So this is the lumen. This is the, you can say, tubular fluid. This is the peritubular fluid. Now, you have a secondary active transport here, transmembrane protein is taking in 2Na plus with 1 glucose. Glucose is exiting via facilitated diffusion. This is exiting via, say, 2K plus, Na plus K plus ATPase. ATPase. Okay. Now what is happening is, if a person is non-diabetic, glucose will keep on getting reabsorbed. Okay, completely. There is no glucose in the urine. That is a pathological medical issue that you should be medical fact that you should be aware of. There is no presence of or there is absence of glucose in the urine. If at all there is presence of glucose in the urine, that specific disease, that is a disease then. After urinalysis or urine analysis known as urinalysis, if glucose is found to be present in urine, that is known as glucosuria, presence of glucose in the urine. And why is glucose completely reabsorbed is, you have a specific concentration of glucose. In the blood you have 70 to 110 mg per dl. That is the normal glucose level. But the kidneys can absorb glucose till a specific level of, you can say, 198 mg per dl, till, you can say, almost 198 mg per deciliter, meaning that the transport maximum, that is known as the transport maximum, okay, means how much glucose can be taken or can be absorbed by the kidneys or by this transporter, okay. If the level of glucose is 198 mg per dl, till that level of glucose, the glucose can be absorbed. If the glucose is till that level, also means you are highly diabetic, it will be absorbed by the kidneys. Okay, so the transport maximum Tm for this specific transporter, for every transporter there is a transport maximum, is equivalent to, you can say, 198 mg per deciliter for glucose concentration of glucose concentration in the blood. That will be filtered out through the, you can say, glomerular capillaries. It will leak out through the podocytes, the peduncles and the filtration slit, and then in the filtrate it will be present, and then it can be reabsorbed. If at all the glucose level increases, rises way beyond 198, suppose it is 300, okay, thereby it has reached the its own transport maximum, okay, level of 198, and it has surpassed it to, thereby the rest of the glucose molecules, okay, means 102, you can say, if you take it in moles, so that will be thrown out through the urine, and that may cause infection in the urine, because microorganisms, they tend to grow in conditions where carbohydrate source is present, where glucose source is present, because they depend upon glycolysis heavily, because they have mesosomes as the, you can say, mitochondria. They won't have real mitochondria, so they depend on glycolysis heavily, right? So the microorganisms and the, which are the prokaryotes, they delve on this, and it can cause urine infection and all. So that is known as glucosuria. Okay, so glucose is completely reabsorbed. That is the answer. And coming back to the next question, proximal and distal convoluted tubules are part of. They are part of nephron, not vas deferens, not oviduct, not seminiferous tubules, because a nephron has the following parts. You will be having the renal corpuscle, okay, that is the glomerular capillary, and glomerular capsule slash Bowman's capsule, capsule, then renal tubule, and then renal tubule you will be having proximal convoluted tubule twisted, then loop of Henle, then you will be having distal convoluted tubule. Okay, this PCT, DCT. In this question you have distal convoluted tubule, DCT. So DCT is very much a part of uriniferous tubules of a nephron. So you have 1.25 million nephrons in each kidney, you can say, million nephrons in each kidney. Okay. So come to the next question. Breast border is a characteristic of. Very, very important question. When you talk about breast border, question number 39, it is a characteristic of which one? It is a characteristic of the proximal convoluted tubule. Know it very, very well. So the thing is, the proximal convoluted tubule, okay, they contain the microvilli, that is known as the breast border epithelium, that is known as, okay. So cuboidal cells with microvilli are present in PCT. I'll just give you a table here. Just know it for a fact. If you talk about PCT, cuboidal cells plus microvilli means which have microvilli, and those microvilli appear as breast border. They have a breast border appearance under the microscope. Okay, breast border cells that is also present in the small intestine. Okay, same microvilli. Now the nephron loop will have both squamous and cuboidal cells. Okay, nephron loop or loop of Henle, the thick ascending limb, if you check, okay, the thick ascending limb has the cuboidal cell, thin has squamous epithelium. So cuboidal epithelium and squamous epithelium. Both are simple, so none are stratified. Okay, all are simple only. Are present in, you can say, loop of Henle. Then if you talk about the distal convoluted tubule, then it may have some microvilli, but in NEET question you will take on PCT because distal may have some, that is in research, but if they have, they will be having with very few if any microvilli is present. Okay, DCT mostly no microvilli as per the NEET syllabus. Okay, but the current research does show that they can have few microvilli in some cases. Okay, so for this question we'll take on PCT only. Okay, we'll take on PCT only. Then coming on to the collecting system, okay, which will have the late DCT, the intergrated cells and principal cells. Both will be cuboidal cells, okay, collecting system. In many books it is known as late DCT, okay. The principal cells and the intercalated cells. So both these cells, okay, have cuboidal epithelium, okay. Cuboidal cells, okay. And then we come to the end, that is the papillary duct, which has the columnar epithelial cells now. Now papillary duct, they have columnar epithelial cells. Okay, this collecting system is also known as late DCT in some old books. Okay. So coming to the next question. Reabsorption of useful substances from the glomerular filtrate occurs in question number 40. So maximum reabsorption occurs in always proximal convoluted tubule, but sometimes it occurs 20 to 30% reabsorption also occurs in loop of Henle. Distal doesn't reabsorb. Okay, collecting tube doesn't reabsorb. Okay, distal mostly will secrete. So you have something known as, in case of filtration, okay, filtration can be divided into three parts. One is glomerular filtration, again another one is tubular reabsorption. This is a very fundamental and pivotal step. Another is tubular secretion. Under secretion, we'll be secreting the hippuric acid, the creatinine, the uric acid, the urea, right? Glomerular filtration, podocytes and all, they will filter, right? Net filtration pressure that you had learned about, that is your glomerular blood hydrostatic pressure minus, you can say, capsular hydrostatic pressure plus BCOP blood colloid osmotic pressure, which came around 10 mmHg, right? Net 10 mmHg. Then under tubular reabsorption, in PCT you have 60 to 70% of reabsorption, okay, of maximum reabsorption happens in PCT. 20-30% can happen in the loop of Henle. Okay, in the entire tubule, if you take, 99% of the solutes and water will be reabsorbed completely in the entire tubule, PCT, loop of Henle and DCT. Okay, DCT is mainly for the tubular secretion, DCT. Okay, and this one we know that loop of Henle, it does which function? Loop of Henle has a very prominent function to set up the NaKCl, Na2Cl minus importer. It has causes the absorption of sodium and chlorine and magnesium and calcium and little bit of potassium. But the thing is, it sets up the countercurrent multiplication, thereby setting up the countercurrent exchange means it causes more and more rapid reabsorption of water and makes it dilute and conserves water. So this was all about the questions pertaining to the worksheet. The urinary system comprising of the questions of the chapter of called the urinary system, or you can say the also the chapter is known by the name as excretory products and their elimination in NCERT. Okay, so now I think you have been able to comprehend the intricate details of this chapter. Okay, and if you've had any kind of queries in the entire chapter, you can definitely contact me on my personal number. I'll be replying to you as soon as possible because this chapter is extremely relevant and has got a lot of deep and intricate concepts. If you do not know, or if you're not aware of, you're not been able to decipher or comprehend the concept, you could be in trouble while solving any other questions. Okay, so we'll keep on solving these questions, additional questions also via worksheets, and to equip you to have proper conceptual prowess over the concepts pertaining to this specific chapter and other chapters as well. So thanks a lot, and let's meet in the next NEET class.